Let's design a vector based on bacteriophage M13. Now we know that one of the most important things required for any vector is the origin of replication, because it has to replicate from somewhere. In bacteria, a small ORI takes a huge burden off because it's just a small sequence which can do it for bacteria. But when we are talking about phages, replication is much more complex. Phage DNA molecules generally carry several genes that are essential for replication. And in M13, they are pretty much scattered. They are not like bacteriophage lambda that they are at the ends. Including genes coding for the components of the phage protein coat and phage-specific DNA replicative enzymes. Now, if you look at an M13 genome, it's 6.4 kilobase pairs long, and we see these tightly packed genes from 1 to 9. There is only one 507 base pair intergenic sequence in which foreign DNA could be inserted. So only 507, is it any good? Can we really use it? Actually, it's no use at all, because it's a very, very small region to clone into. So therefore, we do an interesting thing. First of all, we put a lac Z gene so that ultimately we can distinguish it on blue and white colonies. And we also introduce an EcoR1 sequence. If you look at this bottom hand image, so if you look at this particular here, basically we do this particular mutation and we introduce an EcoR1 site so that we can ultimately clone our gene of interest. At the moment, we are only exploiting 507 base pair long region, but we'll come to it that how we can use it in a more efficient manner. Okay, now then we introduce a polylinker, and that particular polylinker has a huge number of sites for various restriction enzyme. In this way, we ultimately construct and we insert this polylinker into a LAC-Z gene. In this way, we construct vector known as M13 MP7, but the insert it can take is not huge. Now, the amount of DNA it can take is only from 1.5 to 3 kilobase pairs. This is not enough. Okay, we can clone more than that in bacteria. So why would anybody would like to use M13? To improve upon it, the best strategy is to make a plasmid hybrid known as phagemid. So pretty much अगर आप एक plasmid की एक M13 के सीक्वेंस से M13 के जीनोम से शादी करवा दें तो what you get is phagemid. Now phagemid has some interesting qualities because phagemid has M13 DNA fragment and it has, you can put any sort of restriction, but you can also put an ORI. We can use the ORI of phagemid. In this way, it will replicate very fast. So you can see in this particular diagram that you have basically you can get a lot of single stranded molecules out of it, which are being basically packaged into phage particles. Now let's talk about some insert capacities. So we know that plasmid would like to carry from 0.1 to 10 kilobase pairs, normal plasmids which we use in E. coli. Bacteriophage lambda can do 10 to 20. Cosmids can do 35 to 45. We later discuss cosmid. Phosmids are very similar to cosmids, but they have just, if you know about a bit about conjugation regarding the pili, they can form pili as well. That's why they are known as phosmids. And bacteriophage P1, which is another bacteriophage pretty much like lambda, but it has a huge carrying capacity that it can get 80 to 100 kilobase pairs of insert. In this way, we have bacterial artificial chromosome, which has a huge carrying capacity as well, 50 to 300. And then we have P1 bacteriophage derived artificial chromosome, which is 100 to 300. Then we'll move on to yeast artificial chromosome, which is 100 to 2000 kilobases, and human artificial chromosome, which can be used in human cultured human cells, which is greater than 2000 kilobases. But the last two comes under eukaryotes. So we'll discuss it when we are discussing manipulation of eukaryotic cells. But the point which you really need to understand is that there are certain sequences which needs to be there to basically make successful vectors. And despite origin of replication, what we need is an antibiotic selection or some sort of colored selection. And also, if you are going for an expression, then promoter, ribosome binding site, and a termination sequence to basically ultimately make your protein of interest.